All right, very happy to be here again. Give you a little bit of an update. Last year I presented the gene therapy results from the study that we published in 2024, along with a few updates. And today I'm going to talk a little bit about the next steps on that, and also a new variant of AAV9 that we're bringing into play. So, again, just to recap, not going to go through the data, but we had conducted a pretty comprehensive gene therapy study with AAV9, just plain Jane AAV9, injected intrathecally, and we saw these benefits treating newborn mice, and then those benefits diminished as the mice got older. And then we had updated that, and this is unpublished, where we saw that that if we went into the homozygous knockout mice, then as we moved out of the P0 neonate, ICV injection, then we were no longer rescuing the EEG, but we could still partially rescue nesting behavior, and that was about it. Whereas if we stayed in the heterozygous mouse model, then we could rescue the EEG patterns pretty well, all the way out to 15-day-old mice, and we didn't test older than that. Okay, so all of that was using intrathecal, the approach was doing intrathecal administration of AAV9. That's, what I'd say is the current platform for CNS gene delivery. There are other approaches, but that's something that we've used pretty extensively for various diseases that we've moved into clinical trials. All of these are treated basically with the same approach. Similar overall doses, trying to escalate up to about 1 × 10^15 vector genomes per patient. Taysha Gene Therapies has taken Rett syndrome forward for that, and they're about 2 years out on their first patient and reporting encouraging clinical trial data. And across the field, there's now dozens of clinical trials that are using that approach of AAV vectors, mostly AAV9 injected into the CSF. So again, I'm kind of saying this to establish that this is sort of the standard platform right now for doing gene delivery to the nervous system. And so what we were initially trying to do is build upon that platform, and we did a screen here of doing targeted mutagenesis of the AAV9 capsid, and then injecting these libraries of new variants into both mice and non-human primates, and recovering that out of about 20 different regions of the brain, trying to look for things that were sort of or had improved CNS gene delivery. And we were screening in both mice and non-human primates to try to get something that would translate effectively across species. And so we selected this, what's currently our top variant is something that we're calling FYHR, F-Y-H-R, and this was enriched in both mice and non-human primates. It has a couple of point mutation variants on the capsid, and we've shown that it still purifies, it produces equivalent to AAV9, purifies equivalent to AAV9. And so just really quickly, we took an approach where we barcoded, we had reporter constructs that we barcoded. So it's exactly the same reporter constructs, just with a different UTR barcode. And we mixed those together, AAV9 and this AAV9 FYHR, in equal amounts. We injected them together in the same animals, in non-human primates and in mice. And then so that allowed us to do a head-to-head comparison all within the same animals. So if you're doing experiments in like non-human primates, for example, there's huge variability animal to animal. But by doing this approach where we mix the capsids and put them in the same animal, then we could reduce that variability. And so one of the things that we looked at is the cell type specificity of AAV9 by this route, and this is a lumbar intrathecal injection. And then we're using a in situ hybridization approach called marefish to look at RNA expression and correlate it with cell type specific markers. And so we were able to get pretty good and fine level quantification of the different cell types that are being transduced, and basically showing that AAV9 and this AAV9 variant fire are transducing, basically the same mixture of cell types. So it's retaining the same cell type specificity as AAV9. That was important for us. And then if we looked at the biodistribution and the increased efficiency, this is all from mice, again, where these are mixed and injected in the same mice. And we see at the vector genome copy number level, about a 2 to 3-fold increase in efficiency, throughout the brain and the spinal cord. And then we go to peripheral organs, we're losing some of that, although there is increased delivery to the liver. So it's a relatively modest increase, but when we moved to non-human primates, we saw basically the same thing, around this like 3-fold increase in vector genome copy number. And we looked at about 25 different regions across the brain. And basically everywhere that we looked, if AAV9 was high, then fire was a little bit higher. If AAV9 was low, then the fire was low, but a little bit higher. So it followed exactly the same pattern of distribution as AAV9, but everywhere we looked, it was about 3 times higher. That was vector DNA biodistribution. We also did RNA scope to look at expression of the transgene mRNA. And these are just kind of sample images here. Whenever we did this, we were, these are all paraffin sections, so we would cut one slice and look at the AAV9 barcode, and then the next slice over was the fire barcode. And everywhere we looked, it's just there's an increase in number of cells transduced with this variant. And when we quantified this, again, this is some of the data, but the top panels are all different regions of the brain, and then we have like the lumbar spinal cord, nerve roots, dorsal root ganglia. So basically everywhere that AAV9 is going, the fire is going, about 3 times better. So I want to pull that back to SLC6A1, and I will, but the take home here is that we've got now, a variant that behaves exactly like AAV9 if it's injected into the CSF, but it's gonna, at whatever given dose, it's gonna express the gene in about 3 times more cells. So it's basically 3 times more potent than AAV9. Implications of the gene expression, because I just wanted to segue here and talk about this a little bit. So this is the design that we published that was kind of our lead candidate, where we used the MECP2, short MECP2 promoter, the human transgene, and a short poly-A. And I want to highlight that with AAV, you can package this as a traditional single-stranded genome, or you can use a shorter version of this, which your limits are about 2,200 bases of DNA that you can package. And if you do that, you can package as a self-complementary or double-stranded genome. This is important because if you use this compact genome configuration, then it's about 10 times more efficient. So again, if you're apples to apples dose, if you can use this double-stranded DNA configuration, then you can express the gene in 10 times more cells compared to kind of a more traditional version of AAV. So this would be much more potent than a traditional design with a bigger gene expression cassette. Okay. So our next steps here and where we've gone with regulatory strategy, because we published this rescue, we've been engaged with the FDA about how this could translate into humans. And so we're basically at this point where we have the proof of concept efficacy and safety data. We had a pre-IND a number of years ago, and the feedback from that was that we needed to do toxicology studies in rats and non-human primates, and we were going to use AAV9. Now we've gone back more recently and presented all the new data to them and everything, and they actually backed off and said we didn't need to do non-human primates anymore, that they were fine with us just doing a safety study in rats, and they also gave us the green light to bridge everything over to this fire variant, with the understanding that this could be about 3 times more effective. So here's sort of our timeline. We had gotten this feedback from the FDA back in 2020. I say Type D meeting feedback expected in October. We got the feedback during the government shutdown, and they agreed to our plan to do like an abbreviated bridging study to move over to this new variant. They agreed to our request to have kind of an abbreviated toxicology study and not have to do large animals. And we have this bridging study over to this new capsid ongoing right now. And so we've got buy-in from the FDA as far as the plans to do the drug manufacturing, GLP tox studies. And so these next steps, it is possible for these next steps to happen. So with the idea to try to stay on time, I'm going to wrap up and acknowledge my team, especially Ray here, who's been really leading this project after postdoc Francis Schaffo that led this for the first couple of years, and Ray picked it up and has done fantastic work. And then also Matt, who unfortunately isn't in this picture, has really been working side by side with him. And definitely want to acknowledge SLC6A1 Connect for getting us started on this and providing support over the years, as well as Taysha Gene Therapies, which provided pretty substantial funding to this along the way, but is no longer engaged. So with that, thank you everybody, and looking forward to the panel a little bit later.